Hi guys, we're going to talk about photosynthesis in this one, and I'd like to think of this one as the kind of essential concepts. Photosynthesis can be a big topic in terms of details. It is certainly very important, and the ideas in there will kind of test your understanding a lot. So I'm going to focus on the kind of, the essential ideas rather than trying to cover too many details, okay? So let's begin. Now what we need to understand about this is that if we look within a leaf, we're going to have many cells. So, we're going to have lots of kind of plant cells in the leaf there, cell walls, etc. Okay, and within these cells are many organelles, of which one of them is a chloroplast. So if we look within one of these plant cells, we've got the cell wall, nucleus, vacuole, and we're going to have small organelles called chloroplasts. And we're going to have mitochondria as well, but we're focusing on the chloroplast. So let's just quickly look at these chloroplasts, and maybe I could just do this. So if we look at this chloroplast, now what would we see? We would see something like that. Outer membrane, inner membrane, and then inside we have this fluid called the stroma, and within that we have thylakoids stacked up into grana and intergranal lamellae. Okay, and within the thylakoid we have the thylakoid space, or otherwise known as the lumen. Okay, so the photosynthetic process is happening within this chloroplast, and I guess the main idea here is that the two stages of photosynthesis are happening in the two different locations. So here in the membranes of the thylakoid, so thylakoid membrane, that's where the light dependent stage is occurring. Okay, so the light dependent stage is happening in the thylakoids, and even though I'm not going to go too much into it, it's kind of why we have this structure for the thylakoids, so to increase the surface area of the membrane that contains all the stuff that's needed for photosynthesis. So the more membrane we have, the more of the light-dependent stage can happen. Okay? So the light-dependent stage is occurring in the thylakoid membrane, and it makes some products which are then used for the Calvin cycle, or the Calvin-Benson cycle, which is the light-independent stage, which is required. Actually, we'll look into the detail of that later, but essentially this light-independent stage is happening in the stroma. So in the stroma we have the light-independent stage. Okay, so the light-dependent stage occurs, that provides some stuff so that the light-independent stage can happen. The light-dependent stage happens in the membranes of the thylakoids, which make up the grana and the intergranal lamellae, so these bits right here, and that produces the stuff that's needed for the Calvin cycle, the Calvin-Benson cycle, or the light-independent stage, whatever you want to call it, but that is happening in the stroma. Okay, and all of this is a chloroplast right there, sitting in a plant cell, in one of the cells of a plant leaf, for example. Okay, so that's basically setting the scene. We'll now look in more detail. Okay, so we're back in the chloroplast now. Just wanted to kind of take a quick overview of the photosynthesis process, and then we'll look in detail at what's going on. So we have our chloroplast here. I've drawn some thylakoids here. I've drawn this kind of abnormal one, but it's just so that I can show you what's happening inside. So what we have is, we have water that goes into this process. Okay, so water enters the thylakoid, right? And what the thylakoid is responsible for essentially is to produce oxygen. So water goes in, it gets split, and for that process to happen, actually what we could do is draw it like this. So water gets converted, split apart using the energy of light. Okay? Light, you can call that a photon of energy or whatever you want, but essentially it's the energy from light that is used to split the water molecule into oxygen, electrons, and hydrogen, and we are using the energy from the light to produce some other things. So we're producing oxygen, right? Just to keep the numbers decent. So we're producing oxygen, so the thylakoid is responsible for that, but the thylakoid is also responsible to produce ATP and NADPH, otherwise known as reduced NADP. So what we're saying is, the thylakoid is responsible for using light to split water, producing oxygen, but also the electrons and the hydrogen from that splitting are used to produce ATP and NADPH. Now, NADPH is a reducing agent. It can be used to kind of reduce other molecules. That's basically, it's essentially just to modify other molecules, okay? ATP, you should already know that that's a store of energy, so that light energy gets used to produce the ATP, and that ATP can then be used to provide energy for other processes. And those other processes is essentially the Calvin cycle, right? So I hope this makes sense, that within the thylakoid, light is used ultimately to produce ATP and NADPH. That is only half of the story, because remember, what else gets used in photosynthesis is CO2. So let's just show that this oxygen is being produced. Now, just for simplicity's sake, let's just say that 2 water molecules will give us an oxygen molecule. So we're happy with that, right? Water, oxygen. Carbon dioxide, goes into photosynthesis, and what comes out of photosynthesis? Glucose, right? As well as other things, but let's keep it simple, glucose. So we know that glucose has to eventually be produced, okay? But it's not going to come out of the chloroplast yet, all right? So glucose is going to be made, all right? And that glucose is eventually going to be used to make starch in the starch grain. Okay? Now, what's important is that the process of converting inorganic carbon in carbon dioxide, those carbons that eventually get used to make glucose, C6H12O6. So the carbons that make up that glucose molecule, that make up the starch, yeah, that ultimately comes from carbon dioxide. But where do we get all the energy needed to make that happen? Well, that's where the ATP and the NADPH from the light-dependent stage come in, okay? So there's a cycle happening here. Wow, it's a terrible cycle. Okay, so this cycle the light-independent stage, that's where carbon dioxide gets used to make glucose essentially, but that stage will only happen as long as we're providing ATP and NADPH to drive that process. Okay, so ATP will eventually, the energy from that will be used to drive the cycle, and we'll get ADP from that, and the NADPH will eventually come back to NADP, and that ADP will go back to the thylakoid, and that NADP will go back to the thylakoid to get kind of re-energized, as it were. But I hope you can see that it's the products from the light-dependent stage which are used to drive the light-independent stage. Okay? And ultimately the energy that's driving all this is from light. So light allows us, or the thylakoid, to produce ATP and NADPH. The ATP and NADPH then go into the stroma to keep this cycle going. And this cycle is responsible for fixing carbon and making it into glucose. And the glucose is what we store the energy in, and ultimately that energy gets released in respiration. Okay, both within the plant cell and even then, all the consumers of the food chain. Okay, so that's an overview. Now let's look in detail at the light-dependent stage and then at the light-independent stage. Hi guys, so we're back. This is our thylakoid, very simplified, but let's look at our main players. All right, so here we have photosystem 2, made up of accessory pigments and chlorophyll. Okay, so chlorophyll, and we have lots of accessory pigments. Again, we have chlorophyll here again. This is photosystem 1, and again we have just a slight difference between these two chlorophylls. They absorb slightly different wavelengths, but again, that's not the main point that I want to focus on. And again, there's accessory pigments here that increase the range of light that's available for absorption. But again, that's not what I want to focus on. Okay, so we're focusing on how light can be used to generate ATP and how light can be used to generate NADPH. Because those are the two things, the two contributions from the light-dependent stage that will keep our Calvin-Benson cycle turning, and so converting carbon dioxide into glucose. And so without the ATP and NADPH, that cannot happen. So how do we make those two things? So firstly, we start with water. Okay, remember, that's our kind of input, the first input in our photosynthesis bit. Now, what happens is that on the absorption of a photon of light into photosystem 2, so let's say that that's our light right there. So the light strikes the photosystem 2 and eventually makes its way to the chlorophyll. And what will happen is a number of things we need to think of as happening simultaneously, okay? Even though it's probably not actually the case. So what happens when the light energy eventually gets to the chlorophyll? What happens is that energy is used to excite some electrons in the chlorophyll, and that starts a chain of events that will eventually lead to oxygen splitting from the hydrogens. Okay, so instead of H2O, we've now got O and H and H. Okay, and so there's 2 electrons within the photosystem 2 that get excited. So that energy excited these electrons in chlorophyll so that now they are at a high energy state. All right, this is important. So we've excited these 2 electrons in chlorophyll and they have gone up to a high energy state thanks to the energy from light. So we've excited the 2 electrons in chlorophyll, they've gone. we now need to replace them, and the place we get them from is from the hydrogens. So we can split the hydrogens up into protons and electrons. So each hydrogen splits into a proton and an electron, a proton and an electron. And these two electrons can then replace the ones that were lost in the chlorophyll. So essentially what we've done is we've split the water. So if I can redraw that, the way I would do that is I'd show that we have H2O and that's getting split and providing the electrons into the chlorophyll, and we come out with oxygen and we come out with 2 protons. Alright, so 2 protons right there. I hope this is making sense. So light caused chlorophyll to lose 2 electrons, which got raised to a high energy state. And to replace those electrons in the chlorophyll, water was split into oxygen and protons, and those 2 electrons were replaced in the chlorophyll. Okay, so what happens to these electrons that were excited into a high energy state? Well, remember now, the energy from light is now in those electrons, right? They've been raised to a high energy state, and they get passed through electron acceptors and electron carriers. Okay, so the high energy electrons are now moving through. Alright, the electron acceptors, electron carriers, essentially an electron transport chain. Alright, but at this point, the energy in the electron is actually used to do something. And what that thing is the transport, the active transport of protons from the stroma into the thylakoid space. Okay? So, because the energy of these electrons was used to move protons from the stroma into the thylakoid space, the energy of these electrons then goes down. Okay, so the electrons then lose their energy and kind of go into a low energy state. Okay, because the energy was used to actively transport H+ ions or protons from the stroma into the thylakoid space. Now, because the concentration of protons is high here already, right, it's against the concentration gradient, so it's active transport. Okay, so energy was needed, and where did the energy come from to do that? From the high energy electrons. So as they moved through the electron acceptors and electron carriers, as it goes through the carriers, its energy is used up to pump the protons into the thylakoid space. So these electrons are now at a low energy level, and they arrive in photosystem 1. Okay? System 1. But what we need to mention here, and we're going to take a kind of sidetrack here, is, well, why did we bother to pump this proton into the thylakoid space? What was the point of that? The point is that we have a vested interest in building up a proton gradient. Okay? Because as these protons build up in concentration, we build up a concentration gradient. And we build up what is called or a proton gradient, and the reason for that is that as these protons build up in concentration, they diffuse out of what is called an ATP synthase. So this protein is 2 things. It's essentially a channel through which protons can diffuse out, but the kinetic energy of the protons moving out is used to convert ADP into ATP, right? So ADP is combined with a phosphate group, a third phosphate is joined on to make adenosine diphosphate into adenosine triphosphate. And adenosine triphosphate is it's a high energy molecule, which you can break down later to release the energy to make something else happen. Okay? So this is important. So essentially what we're doing is we're using the energy of the electron here to move protons into the thylakoid space. The protons will then diffuse out because you've created that gradient. And we use the energy in that process to drive the production of ATP, right? So you might see in your textbook somewhere that, by moving a proton in, we're going to use that to synthesize ATP. Well, the way that happens is, once we generate that proton gradient, it might even be referred to as, like these protons, as they move out, that process is called chemiosmosis, right? And by moving lots of protons into this area through this electron carrier, we're generating a proton motive force. So this proton motive force, or chemiosmosis, is essentially a way to say we're building up a hydrogen a proton gradient here, so that when these protons diffuse out of this ATP synthase, we can use that energy to drive the production of ATP. So every time proton moves out, ATP is made. Okay? So that's the point of that. Now let's go back to and see what happens to these electrons. So we said that in moving H+ ions into the thylakoid space, the electrons lost their energy, but then they entered photosystem 1. Now that's our clue. Photosystems, they absorb light and they do something with that light energy. And in this case, what we do is we raise the energy level of these electrons again. Okay? So we raise the energy level of these electrons again, and maybe I'll just draw another protein here. A protein called Nadp Reductase. Nadp Reductase. And I don't think you need to know the name of this, but it's just there so that I can explain what happens to these electrons. Okay, so photosystem 1, electrons have been raised to a high energy level again with the absorption of more light, and they pass on to a protein called Nadp Reductase. And you don't need to know its name, but the job that it does you do need to know. And what it does is it takes these high energy electrons and combines them with NADP. Okay, so these high energy electrons, they raise in energy levels, they get combined with NADP and protons from the stroma. Okay, so what do we get when we combine protons and electrons? What did we get when we split the hydrogen atoms? When we split them, we got electrons and protons. So if we recombine some of these protons that have diffused out with electrons from the photosystem, we're going to get hydrogen again. So NADP combined with electrons and protons gives us NADPH, or otherwise known as reduced NADP. Reduced NADP. Okay, and remember that was the other thing that we needed to generate from the light-dependent stage. So the observant of you will have noticed that we've already, in this process, we've already got the two things that we needed to have. We've used light, we've split water, we've generated ATP, we've also produced some oxygen, and we've also got our NADPH. All right, so what I'm going to do at this stage is I'm going to do this. All right, I'm going to highlight the two important products of this process. We've produced NADPH, we've produced ATP, and we've seen how that happens using the energy of light. Okay? Now, to save time, I'm going to not go over things again like I like to do, but just ask you to maybe rewind and look at that again if that's not making sense. Okay, I can't resist the temptation. I am actually going to go over it again. So, let's do that. Light was absorbed in photosystem 2. Chlorophyll's electrons got excited and left. Okay? These electrons were replaced by the splitting of water into oxygen, 2 protons, and 2 electrons. So the 2 electrons went to the chlorophyll, and chlorophyll is now, you know, it's replaced its lost electrons basically. But the electrons that were raised in energy level, they are now up here, and they pass through a series of electron carriers and electron acceptors. In that process, hydrogen ions are pumped into the thylakoid space. Our electrons now lose the energy because their energy was used to actively transport protons into the thylakoid space. The hydrogen ion concentration increases, the proton concentration increases in the thylakoid space. This generates a proton motive force as protons move out of the ATP synthase by diffusion. As they diffuse that back out into the stroma, we use that energy, that kinetic energy of those protons, and we use that to make ATP. So we've made ATP. So what's happened to these electrons? They've passed to photosystem 1. We now absorb more light, and the energy level is raised again and passed on to, these electrons are now passed on to Nadp Reductase, which combines those electrons with NADP and uses 2 protons that are in the stroma because of the diffusion out of the thylakoid space and generates NADPH. Okay, so main point, light absorbed, electrons excited. We've used the electrons' excited state energy to essentially make ATP, but we've also used those electrons to make NADPH combined with some protons. Okay, and essentially that's the main point of the light-dependent stage, to make ATP and NADPH. Okay, there is one alternative that we need to be aware of, which is sometimes, and this is the way I like to think of it, and you might see some other conflicting evidence, but you know, I won't tell you anything that's going to damage you. In fact, it might just give you a kind of logic as to why some of these things happen. If there isn't enough NADP to accept these excited electrons, we don't waste that energy. If there's not enough NADP, these electrons who have just kind of, you know, been raised up again, their energy level's gone back up. Do we waste that energy? No, we don't. If there's not enough NADP to go down this route, what we do is we send those excited electrons back to the electron carriers. They pass back through the electron carriers. Again, more protons are pumped in, and the electrons lose their energy again in that process and return back to photosystem 1. So, what I'm saying is, if we don't have NADP, we don't waste that energy in those excited electrons at this stage. What we do is, we say, all right, we can transfer you back to the electron carriers, we can use your energy to pump more protons in to the thylakoid space. And we keep going round and round again. Okay? And in that process, we build up the proton gradient, and that proton gradient is ultimately used to form ATP. So in your books, you might see that this stage, this thing is called cyclic photophosphorylation, and the reason is that the energy from light is used to excite the electrons, i.e. the photo bit. Those electrons are then used to pump protons into the thylakoid space. The gradient created by the pumping of the protons is used to make ATP, i.e. cyclic photophosphorylation. Okay, so that's that. And yeah, so, and in contrast, when we are not using this cyclic route for the electrons, i.e. when there is enough NADP, the electrons actually go to the NADP, and the flow of electrons essentially is from photosystem 2 through the electron carriers to photosystem 1, through the NADP reductase, and to NADP. This, i.e. makes like a linear route for the electrons. So photosystem 2 loses some electrons, this is replaced by the electrons in water, and so this is a kind of, the electrons just go through to NADP. This is called non-cyclic photophosphorylation, because again, the energy from light is used to generate the proton gradient, which is used to then make ATP. So this is called non-cyclic, but when we don't have NADP, and essentially the electrons from photosystem 1 are circling between photosystem 1 and the electron carriers and back again, absorbing light, using the energy to pump protons, going back to photosystem 1, absorbing light, going back to the electron carrier, pumping protons, losing energy, and so on. That, as you can see, is the same electrons going round and round. That is called cyclic photophosphorylation. Okay, so let's move on. Let's look now at how ATP and NADPH are used to essentially convert carbon dioxide into glucose. Hi guys. So here we're looking at a very simplified version of the Calvin-Benson cycle. Okay. In one of my playlists, I've posted a video of a BBC documentary about how this kind of was figured out, how the stages of how essentially photosynthesis was figured out, through experiments and investigations done throughout history, and that's quite interesting. I'm not going to look at that. But the fruit of that knowledge is basically here now. So let's look at how we convert carbon dioxide into glucose. Glucose being a hexose sugar. So maybe, for example, we can use glucose right there. Okay, so essentially, how do we convert something inorganic like carbon dioxide, and how do we make an organic molecule out of it as a store of energy, as the basis of so many things? Amino acids, nucleic acids, carbohydrates, structural carbohydrates, cellulose, energy storage carbohydrates, starch. It all comes down to this cycle. Yeah. So, and this stage right here, the conversion of inorganic carbon into something organic that then forms the basis of our biological molecules, that is a really important step. Okay, so right, let's have a look now at this. So now remember, this is happening in the stroma. Okay, and so let's begin. So carbon dioxide comes in from the atmosphere, diffuses into the leaf, through all the fluid cytoplasm, and then eventually makes its way into the chloroplast, into the stroma. And in the stroma, it will encounter an enzyme called ribulose bisphosphate carboxylase oxygenase. I don't think it's necessary, but it's got a shorter name called rubisco. Okay, but what it does is it combines that carbon dioxide with a pre-existing molecule called ribulose bisphosphate. And what that does is, and this molecule has 5 carbon atoms in it. And what's important to know is that when it combines carbon dioxide with this 5 carbon molecule, we produce 2 times 3 carbon molecules. All right, so we are just accounting for the carbons here. So, and you don't need to know the structures of those molecules. You just got to be able to appreciate what the relationship between this and the previous stage is, and secondly, just to be able to account for the production of glucose at the end and the maintenance of the cycle. So, carbon dioxide is combined with ribulose bisphosphate by this enzyme. And what that enzyme does is combine those two and makes essentially a 6 carbon molecule, which then is converted into 2 3 carbon molecules. Okay? Now that glycerate 3-phosphate is then converted to triose phosphate. Okay? This is a 3 carbon sugar essentially. Okay, so this is also 3 carbons, but there are other modifications happening that we can't see. We're just kind of looking at the number of carbons, but essentially we convert glycerate 3-phosphate into triose phosphate, and it's this triose phosphate that can be combined to form glucose. Okay, so 5 carbon molecule combined with carbon dioxide by rubisco, it produces glycerate 3-phosphate. Glycerate 3-phosphate is then converted into triose phosphate. Triose phosphate is a 3 carbon molecule that can be combined to form glucose, and so we have it, carbon dioxide to glucose. Remember, there'll be an enzyme that carries this process out, there'll be an enzyme that carries this process out, and there'll be an enzyme that carries this process out. This is important for you to understand. You don't need to know the names of those enzymes. Okay? But main point being, we have made glucose. Okay? Now, here's the thing. Why do we need the light independent stage? So why do we need the light dependent stage at all then, if this is happening? Well, for 2 reasons. First, this step right here needs a lot of energy input, and it also needs a reducing kind of molecule, i.e. it won't happen without the energy from ATP, and it also won't happen without NADP, NADPH, the hydrogens from that molecule either. Why do I do that? Okay, so NADPH is converted back to NADP. The H part, the reducing part, is incorporated, is needed in that conversion, and the energy from ATP is also needed in that. So, without this, without the ATP, without the NADPH from the light dependent stage, this part of the cycle would not occur, and without one part of the cycle, we wouldn't get it continuously being able to turn. Okay, so that's one thing. The other thing is, we could have a situation where the carbon dioxide is used up. So the ribulose bisphosphate is combined with the carbon dioxide and it's all used to produce glucose. Well, if that happened, we wouldn't be able to continue to fix any carbon. The process of combining inorganic carbon dioxide into organic molecules being known as fixing, we wouldn't be able to fix any carbon dioxide in this process if we didn't somehow regenerate this molecule, this 5 carbon molecule, ribulose bisphosphate. Okay, so what we need to know is that whereas some of the triose phosphate is used to produce glucose, some of it is also then used to make sure that we regenerate ribulose bisphosphate so that we can combine it with more carbon dioxide. Okay? Remember, enzymes are very specific about their substrates. If we run out of ribulose bisphosphate, it's not like the enzyme can just go and find something else. It's only complementary to ribulose bisphosphate and carbon dioxide. Okay? So, yeah, and I guess the other important thing is that even this step needs ATP. Okay? Alright, so there we have it. Okay? This stage would not happen without ATP, without NADPH. Alright? So we need this cycle to keep, going in order to take carbon dioxide from the atmosphere, in order for that then to produce glucose, that will then be converted into starch, other things, glycerol, yeah? And I believe at this stage we get things like fatty acids can be produced from this right here, amino acids. Okay, so this cycle is essentially the starting point to make so many biological molecules. It's essential that it keeps going. And the way we do that is by regenerating ribulose bisphosphate, not using all of this up to make glucose. Okay? One last thing. This stage right here is called fixation. Okay? So okay, this is called fixation, incorporating carbon dioxide into an organic molecule. This stage right here, glycerate 3-phosphate to triose phosphate, is called reduction. Okay, because we're using the reducing power of reduced NADPH. Remember, reduced NADPH is going to turn into NADPH. Where has the reduced gone? Well, it has to pass it on to something. It's passed it on to the glycerate 3-phosphate. Okay? And finally, the conversion or the use of triose phosphate to regenerate ribulose bisphosphate is called regeneration or reformation. Okay, and that's that. Okay, so remember, this ATP, this NADPH, they are making sure that this cycle is turning, right? And they were produced in the light-dependent stage. And that's the essentials of photosynthesis. I think we're done. Good luck, guys.